We have yet to find any place on Earth that does not have life. There are places where there are not many plants and animals, but when you look at the Earth from this side, I like looking at the Earth from the Pacific Ocean because it sort of takes away the anthropocentric view that we often have of life on Earth and really drives home the point that most of life on Earth is microbial. And for the 150 years that we've been able to grow microbes in lab, we asked them to do an extraordinary thing, which is get one cell by itself and say, divide every hour, like a bunch, and then turn into a colony and we'll study you. And it's extraordinary because if every cell on Earth did that, they would suddenly make a whole nother Earth in not too long of a time. So how we start to learn about these microbes is by what they breathe and what they eat. So we, of course, are pretty simple. We can only breathe oxygen, and we can only eat a bunch of organic matter, and not all that much, many different types of carbon that we can eat. But microbes have been on Earth for 3 billion years longer than we have, and they can breathe things that read like a periodic table. They can breathe arsenate, perchlorate, oxalate, literally electrons coming out of batteries directly. If you gave them a pizza, they could eat the pizza and the box that it comes in. They can eat everything in the lake, but that's what we know about from cultures. But suddenly, or in the 1990s, we developed the ability to sequence DNA and realized that the cultures were the tip of the iceberg. Everything is even more complicated than we thought. So now we can actually go into the environment, chemically pull out DNA, and read it like a book. And now we have a big library of books. So our first thought when we first started being able to do this is, sweet, these things have never grown on a petri dish. Let's find out what they want to eat and then feed it to them. And they'll do this thing where they magically divide every hour and they'll grow into colonies, and we can include them in our studies and learn lots of stuff about them. So one of the first studies that we did was to go out into the Pacific Ocean off the coast of Peru in this big drill ship that is used solely for scientific purposes. It's one of 2 in the world. And I made a box for every DNA sequence that we got, and I colored it by how closely related it was to something we knew before. And they're all white because they're totally not related to anything. They're totally new stuff. We call them microbial dark matter. So now we tried to culture things, thinking, okay, these are new stuff, we'll get some cultures. Well, the cultures are dark blue. They look just like what we had before. There was nothing new in the cultures, and they don't line up. We didn't get anything that we got in the DNA sequences. So suddenly, we realized that this was a really big problem, nearly intractable problem, and these things are like aliens on Earth, and they truly are. Even this is not that exciting of an alien because it still has like 2 eyes and a head. But microbes could be anything, and what we need to do is to meet them where they are. If they're refusing to grow for us on a petri dish, then what we have to do is go as far as we can with their DNA and their RNA and the stuff we can get. So what we can do is we can put them on a tree, because the first thing we want to know is, are we related to them? How closely related are we? Raise your hand if you learned the 5 kingdoms of life theory, and maybe you still think it's true. It's totally wrong. You're totally wrong. So Monera is that one that used to be one thing. Animals, plants, fungi, protists, that's all the same thing. But now that we can do DNA sequencing, we know that it's not a tree of life, it's a big bush. And I love these moments when nature says, yeah, I see your classification systems, and I don't care. I'm going to make things in between. So our classification systems are thrown completely into array by all these novel, deep evolutionary branches of life that really, we don't know what they do, and we can't figure out much about what their genomes do either. This is on the y-axis here, we have the percent of a genome that we can figure out what it does. For things in culture in the petri dishes, we still have some stuff we can't figure out, but for the uncultured things that we're just getting DNA from, we have a much higher content of parts of the genome that are just dark to us. We don't know what's in them at all, which sets me up for a really fun career, because my job is to figure out what they're doing, and I get to study them here. So I recently, I have projects sort of all over the world, but 2 weeks ago I just came back from Costa Rica, and so you can see there's a subduction zone right off the coast of Costa Rica. So that trench right there is where the oceanic slab is getting pushed underneath Costa Rica, and as it goes down, it makes all this water come up in the forearc, and then the arc is running through the middle of Costa Rica. It's where the volcanoes come up, and this is all driven by subduction, and you can see pins where we all visited. So we're looking at the microbes that are there and that are living on these deeply sourced fluids. So this is me standing in the jungle with some of the rest of our team. You can see this orange schmutz in the water. Those are the microbes that we're studying. We have no idea what they are. That's what my grad student is working on, hopefully right this minute, if I'm lucky. That's her. That's Kate Fullerton, and she was awesome. She packed for this entire trip, having never been in the field before, with her absentee PI not helping very much except by email. And she's filtering water. We did a ton of filtering water to catch the microbes in the filters. And there's more of this orange crusty stuff that we're going to figure out what that stuff is. This trip culminated in a hike down into the Poas crater. Poas is an active volcano. This thing explodes once or twice a year, and there I am with my Italian colleague Donato, and we're standing in these sulfur vents come up, and I was asking our Costa Rican volcanologist, what do we do if it erupts? What's our exit strategy? He said, turn around and enjoy the view, because it's your last one. So I also have work in the Arctic. It's 79 degrees north in Svalbard. It's not really Norway. Norway kind of administers it. Russia kind of has a part too. But you can see in this glacier, there's a bunch of red stuff coming off. I don't know if you can see that. That's iron-rich hematite that's coming off the glaciers and is feeding this really kind of strange microbial ecosystem in the fjords. And so there are these lovely bacterial blooms. Those white things are not snow, but microbes that are growing in the next to the glaciers. That polar bear was a mama that we saw from far off with a baby, and that is a terrifyingly large paw print, but we were safe. She was far away when she was actually there, and it's just a lovely place to work. And I'm grateful both to my graduate students and to funding agencies who are so enlightened as to realize that you have to chase these uncultured microbes to figure out what's really happening on Earth. Thank you very much for listening.